welcome Miss Simran Motkekar to our show that is Rendezvous with Scientific Minds. First, I would like to introduce you to our show. This is Miss Simran Motkekar. She has completed her bachelor's in zoology under Mumbai University, and further she has completed her master's from SIS College Science. Her certifications are she has successfully achieved summer training program in microtomy, which I was held in VG Vazay College, and much more accolades to her. Over to you, Miss Simran. So, can you
Thank you.
tell what topic we will be discussing? Isn't it interesting?
Yes, it is really interesting, and it is the most common, mostly studied pathway that we are going to
Oh,
discuss today. Yeah.
that means the students are going to get a lot rich content from glycolysis today, don't you think?
Yes, yes, and the speciality of this video would be like it would be much more in simple form
Hmm.
rather than a deeper one, so that people find it hard to recollect or to study that.
Study about.
That's why.
Before starting with our topic, let us just do some generalized discussions about it.
Yes.
So, what do you think glycolysis is?
Glycolysis is a process from where energy is produced,
Mm.
which is utilized all over, oh, which is utilized by our body.
Mm.
So it is the major pathway in the body.
Major pathway. You mean to say whatever food we are consuming that is being synthesized, that is
Yes,
being used up by our body, correct?
yes, yes.
So, can you put it in simple terms like in just one word or something like that?
Uh, it is energy synthesizing process, we can say.
Yeah, energy synthesizers, right? Awesome.
Yes.
So, what are the pathway all about? Sure, I'll just start sharing your presentation right away. We'll
Yes.
start from that. I hope the screen is visible.
Yes.
I'll put it in slideshow. So, since we were discussing glycolysis, let us understand what does this mean.
Yeah, before actually starting glycolysis, I would like to bring up few points about it.
Yeah, sure.
Glycolysis comes under carbohydrate metabolism. It is the first and foremost pathway that takes place.
Mm-hmm.
So, what is carbohydrate metabolism? If the question arises, then the answer is carbohydrate metabolism is mainly the food particles, sorry, the food we eat that
Mm.
is broken down into various particles,
Mm-hmm.
and that is lipids, amino acids, carbohydrates. So, these all the molecules need to be synthesized further so that energy can be
Used up
generated,
from our
used
body.
up. Yeah,
Yeah.
yes.
Like,
So,
for instance, if you take like carbohydrates, what
yes,
when we do the breakdown of this term carbohydrate, like it basically carbon, hydrogen, and oxygen, right?
yes, yes.
So the mixing and matching of these elements, we can say.
Yeah, we
Hmm.
can say that.
Yeah.
Yes,
So let us go into the deeper meaning of what does this mean, why this name glycolysis is.
yes, yes. Yes.
Oh,
So the name itself suggests that is glycose means sugar and lysis means breakdown or splitting.
okay.
So larger molecules of sugar are broken and simplified further into simple molecules or macromolecules.
Mm. Like if you say if we are studying something a difficult concept or something like that, what we do, we break it down into segments and then we try to study the small segments first.
First,
Somewhat like
yes,
that our body
yes,
does.
yes, yes,
Yeah.
yes, yes. Yeah, the same procedure we as we do while studying.
Yeah.
It is the same. Yeah.
That's kind of funny. Analogy it was. Should I move to next one?
Yes.
Please let me know when you want to move to next one.
Yes.
Oh, who are these people?
Yeah. These are the scientists Embedden, Mirov, and Parnas, the next scientists of Parnas.
Okay.
So these are the people, sorry, these are the scientists who have majorly contributed in finding out this glycolysis
Glycolysis.
pathway. Yeah, they
Gustav
are major
Emden
studies.
and Otto Meyerhoff. Okay.
Yes,
What are
yes.
where are these people from? Where are these scientists are from?
Uh, uh, no, I haven't gone that
Okay,
deeper.
okay, it's all
Yeah,
right, no problem. So
sorry.
it's written Gustav Emden. It's also called as Emden Meyerhoff Parnas pathway, right?
Pathway. Yes, yes. EMP pathway. So
Okay,
embedded.
no problem. Okay, there are 10 steps it is written. Can you explain it?
Yes, so glycolysis takes place mainly in cytosol of the cell, and it occurs in every part of the body.
Mm-hmm.
So it is a 10-step process that is divided mainly into three phases, we can say.
Mm.
That is preparatory phase, splitting phase, and oxidative phase.
Okay.
In preparatory phase, that we will study further uh, in step by step.
Mm.
But here, the first steps in the preparatory phase, the ATP is consumed
Mm-hmm. Means
and,
it is getting used up.
used up. Yes, and in the oxidative phase, it is the energy is regained. That
Okay.
is reproduced again.
Mm. Okay.
So that
So, yeah, let
yeah,
us go understand the step one by one. We can say
yes,
usually in
yes,
our
yes.
textbooks and conventional method, they just write the flow chart of glycolysis and
Lysis.
expect students to understand that what exactly happens. But just without giving any structure and explaining, it won't make any sense, right?
Yes, exactly. Because
Yeah.
the molecules from where it is going and where it is attaching,
Mm.
that is an important part if you want to study glycolysis.
Yeah, absolutely. And that is
Yeah.
the reason that students get very detached from such topics because they are not exactly understanding from where the molecules are moving and what exactly the molecules are becoming, right?
Yes, yes.
Okay. Can you elaborate in this context, please?
Yes. So this is the first and foremost step of glycolysis, that is uptake phosphorylation of glucose.
Okay. What does this
So
phosphorylation mean?
phosphorylation means addition of phosphate group.
That is
Since
convert like this?
yes, addition
Hmm.
takes place and the molecule changes its structure. So this process is the first step, as I said,
Hmm.
and the glucose, which is in
Hmm.
simple form, gets means the phosphate group gets added on the
Added.
sixth position,
Mm-hmm.
so that it becomes glucose 6 phosphate.
Okay, I'm
And
observing something here that is
yes, yes,
hexokinase. Can you explain what it
that
does?
is the enzyme required
Hmm.
to convert the glucose to glucose 6 phosphate, that is
Hexo
hexokinase
hexo means six, right? And kinase
6
means addition of phosphate. That is why
phosphate.
the phosphate is going and getting added on the sixth position or something like that.
Yes, yes.
Okay. So
Yeah.
I can even observe a ATP getting moved to
ATPs
ADP.
can
So
yes,
that
yes.
means what is ATP?
ATP is adenosine triphosphate, that is getting converted to adenosine diphosphate. So here the one phosphate group that is converting and
Getting
attach
added to the
with
oxygen.
uh, yes, yes.
Hmm. And this
And
Mg is acting as a
Mg,
catalyst or something like that?
no, it is a cofactor
Okay,
required over
cofactor.
here in this step. This
Okay.
is the irreversible step. So
Hmm.
once this step occurs, the
Hmm.
glycolysis pathway is locked and it
Hmm.
has to move further.
Okay.
Yeah.
Okay. Okay. This is a
Yeah,
step two which we were talking about, right? This is isomerization of glucose 6 phosphate. What does
phosphate. Yes.
isomerization mean?
Isomerization means simply converting from aldose to ketose form. As we can see over here, the glucose means the six structure compound is getting into converted into five structure compound, five carbon compound.
Okay.
Yeah, yeah. So again, here the cofactor required is Mg2 plus
Magnesium
glucose 6.
ions.
Yes, and glucose 6 phosphate is getting converted to fructose 6 phosphate,
Okay,
and
we
the
can see
enzyme.
a drastic change in the structure actually. In this six carbon compound, six carbon closed compound is converted
Yes,
into five carbon closed
5
chain
carbon.
compound.
Yes.
Okay, awesome. And what exactly, why this exactly is happening? Wh- What is the role of phosphohexo isomerase?
Yeah, it is phosphoglucoisomerase.
Okay,
Uh,
yeah.
it is an enzyme which helps for the isomerization. The name itself is
Basically, it breaks
isomerization.
the six carbon closed chain to five carbon closed
5
chain.
carbon.
That
Yes,
it does, correct?
yes.
Awesome. Next is
Moving on to the
phosphorylation of fructose 6 phosphate. What does this happen? What exactly happens in this step?
yes, this is the third step in which
Mm-hmm.
again another ATP is utilized over here.
Mm-hmm.
So fructose 6 phosphate gets converted to fructose 1,6 bisphosphate in the presence of phosphofructokinase 1. Again,
It
one
is also
ATP is,
abbreviated as PFK1.
yes, yes,
Okay.
PFK1.
Hmm.
Yeah. So
What
uh,
does bis means?
bis means, um,
Is it like two present
yeah,
in both
means
the
bi
positions or
and
something?
bis. Yes, yes,
It has occupied first and sixth position, right? The
yes,
phosphate.
yes. Phosphate,
Okay.
first and sixth phosphate.
The change is being viewed here very nicely.
Yeah.
Okay. Basically, in this, there is one more addition of phosphate group in the five carbon closed in furan ring, right? Furan only, I
Yeah.
think. Five carbon closed phosphate. Uh, is there any disturbance, Ms. Simran?
No, no,
Okay.
nothing.
Next, can we move to next step?
Yes.
All
This is the fourth step where the fructose 1,6 bisphosphate
right.
is getting converted into means
Two
two
different compounds.
compounds. Yeah.
Hmm.
So fructose 1,6 bisphosphate is a six carbon compound.
Mm-hmm.
No, yeah, and it is converted into three and three molecules each.
Three
So
carbon molecules.
one
Okay.
compound. Yes, yes. That is dihydroxyacetone phosphate and glyceraldehyde 3 phosphate.
Oh. So
And this
which
is,
one will be used in further step?
sorry,
Which compound will be used in further step? And what is the
uh,
relevance of dihydroxyacetone phosphate? And how exactly aldolase works here?
okay, aldolase is a converting enzyme again over here which is used,
Mm-hmm.
and it is an irreversible reversible
Yeah, it's
step we can
reversible.
see there.
Ah,
So glyceraldehyde 3 phosphate will be further used.
okay. And what is the role of this dihydroxyacetone phosphate? Whether it will aid in further reaction or not?
No, it will not, but means it is said that uh,
Mm.
from both the molecules, the further reactions can take place, but mainly it is glyceraldehyde
Mm-hmm.
because since they both the compounds have equal distribution of carbon, so that's why the process goes on further
Maybe this
smoothly.
could happen, right? Like there could be a stage in structure and it could get converted into glyceraldehyde 3 phosphate and then further reaction could take place.
Yes.
Hmm. And then what does aldolase means? What exactly is it that aldol condensation is happening in this step?
Yes, it
Okay.
is.
This step 5. Yes,
Yeah.
Simran, are you with me?
Yes, yes.
Yeah.
Isomerization of dihydroxyacetone phosphate takes place.
Okay.
So, means the
Basically, it is getting converted into
what
glyceraldehyde
wetted
triphosphate.
nitric
Okay.
phosphate. Yes,
Basically, this step is not at all mentioned in our textbooks, right?
textbooks. Yes,
Yeah.
yes. That's why
So
student
just
gets confused.
yeah,
Confused that is it a
how
10
it is
step or a 9
happening.
step? Yeah.
How it is
Mhm.
happening, that we need to understand, right? Like
Understand.
there should be a reasoning for whatever things are happening. Without
Happening.
reasoning, if you give some solution also, it will become a problem for students to understand.
Yeah. Yes.
So this triose phosphate isomerase basically rearranges the structure to glyceraldehyde 3-phosphate, right? And then this
Yes.
G3P will be used further in further steps, correct?
Steps. Yes, yes.
Okay. So what happens in next step? Just
Yeah.
G3P. What is happening along with it?
Yeah. That is dehydrogenation step of glyceraldehyde 3 phosphate.
Hmm.
So, um, here glyceraldehyde 3 phosphate is getting converted into 1,3 bisphosphoglycerate.
Hmm.
So, so there is a production of one NADH, that
Okay.
is nicotinamide adenine
Dihydroxy.
dihydroxy.
Okay. This NADH basically carries hydrogen ions, right?
Yes, yes. So it is an electron acceptor, which
Mm-hmm.
is further studied into the electron transport chain that we can
That
see it
means
later.
in this step, reduction reaction is happening, right? Basically,
Yes,
hydrogen molecules are getting kicked out.
s- convert. Yes, yes.
Awesome. So this mystery is also solved. What exactly happens? Let us
Yeah.
understand the next step.
Yeah. The conversion of 1,3 bisphosphoglycerate into
Hmm.
3-phosphoglycerate.
Hmm.
So over here, 1,3 bisphosphoglycerate is itself a very energetic compound. So
Mm-hmm.
that's why it transfers its phosphate group to ADP.
Mm-hmm.
So ADP gets converted into ATP. So
Okay.
with 3 phosphoglycerate, one ATP compound is formed.
Okay.
So over here,
Mm-hmm.
after this used means consuming, after consuming the ATPs, this is the first step where ATP is reproduced again.
Oh, okay.
Yeah,
That means you're meaning to tell me that this phosphate is getting released from this position. And it two
position.
phosphates are there basically in
Yeah,
this phosphoglycerate.
phosphate.
So we
Yes,
don't
yes.
need two phosphates in this structure. So one phosphate is being
Phosphate is
released, and
is
that phosphate is being taken in by adenosine diphosphate in
phosphate.
presence of
Yeah,
magnesium so that it
sure.
could shield the negative charges there. And this
Yes,
phosphoglycerate kinase basically helps in aiding this reaction to form 3
yes,
phosphoglycerate. Only one in one position phosphate is present, that is 3 phosphoglycerate, and this phosphate is being lost to ADP, that is becoming ATP here, right? As an
yes, yes.
energy production happening in this step.
Yes.
That is some confusing things, right? So much things are happening in this step.
In this step.
And even
Yes,
in textbooks, if you see, there is just a name written, and people are expected to understand, students are expected to understand ki ha ATP is getting generated. That is so wrong, don't you think?
yes, yes. It is wrong because it's really confusing.
Sorry. Hmm.
Yeah, where exactly the ATP is getting,
Yeah.
means how exactly, not where,
Hmm.
how exactly the ATP is again
Hmm.
reproduced in.
Reproduced, or how
Yeah,
come these things are happening that we are able to understand when
understand.
we see the structures. We are not expecting students to draw the structures here, but for understanding point of view they should at
Purpose
least use this
this. Yes,
in
exactly.
textbooks. I feel that, I personally, it's my opinion basically.
Yeah. No, it's actually true. true thing
Hmm.
that this like this splitting of things, if there is there in the textbooks,
Hmm.
then student will really will be benefited to learn the glycolysis, and it is also interesting one.
Yeah.
Yeah.
What is this intermolecular shift of phosphate?
Yeah, means it is again just like I um,
Yeah.
sorry 3 phosphoglycerate again converting to 2 phosphoglycerate. This is a reversible reaction again. So phosphate group's position changes from third to second.
That is because
So
of this mutase, right?
mutase enzyme,
Exactly.
phosphoglycerate mutase. Yeah.
Hmm.
The cofactor again here used is magnesium.
Magnesium. Mutase basically helps in rearrangement of the
Of
structures, right?
structures. Yes,
Yes.
yes,
Uh, Awesome. Next is step 9. We have reached to step 9, Simran.
yes,
Now, can you explain what exactly happens in this step?
yes. The dehydration of 2 phosphoglycerate takes place.
Hmm.
So water molecule
Hmm.
is released, and then
Hmm.
there is a formation of phosphoenol pyruvate.
Okay.
Yeah.
Phosphoenol pyruvate. That
Uh,
enol is because of this double bond which is
double
getting
bond.
added here.
Yes,
That's the
yes,
reason, right?
yes.
And this enol is basically helps in dehydration
Mention.
and
Yes, the
rearrangement of structure.
of the
Enol,
structures.
this ene
Yes,
structure bond is added because of this enolase, right? And we
yes,
need
yes,
phosphoenol pyruvate further. Let us see what happens further.
yes. So here the phosphoenol pyruvate gets converted to pyruvate.
Hmm.
In so again this is the phosphate group energy. Yeah, production step.
Hmm.
So again the second ATP is formed over here.
Hmm.
Yeah.
In presence
So
of
instance of
potassium also
potassium.
now this time.
Yes, yes. So it acts as a cofactor only
Hmm.
in this step.
A
So
pyruvate kinase basically what it does is it leaves the phosphate group from phosphoenol pyruvate, right? And it
yeah,
makes the phosphate to get added into ADP. And this
ADP.
ADP and IP gets added, that leads to formation of ATP. That
Hmm.
means, and in second, this is the second step where energy production is happening, right?
Happening. Yes, yes.
Mm. So this is the overall energy chart, I feel.
Energetic. Yes, it is an
Yeah.
energy chart.
Yeah.
So as we can see that in the step first, the
Mm-hmm.
enzyme hexokinase, in the presence of enzyme hexokinase,
Yeah.
one ATP is
Is
utilized.
utilized. Yes.
Yes. And again in the third step,
Mm.
in the presence of enzyme phosphofructokinase, once
Mm-hmm.
ATP is utilized.
Mm.
And then in the seventh step, in the presence of phosphoglycerate kinase,
Mm-hmm.
ATP is
Is being generated, I think.
generated. Yes.
Mm-hmm.
So it is two ATPs
Mm.
generated, and again here in pyruvate step 10, that is pyruvate in the presence of enzyme pyruvate kinase,
Mm.
again 2 ATPs. So net gain is like 4 minus 2, that is 2 ATP generation.
That means 2 ATP is used
Is
and 2 ATP is
ATP
generated
is
in this cycle.
generated. Yes,
That is balancing the cycle.
it's resigning. Yes.
Okay. Additional step in anaerobic condition. What does this mean?
Yeah, since glycolysis takes place in aerobic as well as anaerobic conditions,
Mm-hmm.
in aerobic conditions, we can see that pyruvate plus carbon dioxide and waters are formed.
Mm.
Same way in the anaerobic condition, pyruvate is getting converted into lactate in the presence of enzyme lactate dehydrogenase. This is a reversible reaction again,
Mm-hmm.
and one NAD is produced, utilization of NADH takes place. Lactate is mainly used by the muscles,
Mm-hmm.
the sports person's muscles. We can find lactate. It is again a source of we can say muscles get energy from lactate in the
Okay.
anaerobic condition. So
Oh,
that's why
yeah.
this takes
Basically,
place.
whenever we do some hard exercises, there is a lack of oxygen in certain parts of our arms and legs, right? At that time, such kind of glycolysis might take place, you're saying, right?
Yes, yes.
Okay. Regulation, what does this mean? Means. Glycolysis means what? Sugar breaking, sugar breakage in our body. So, is there any negative feedback or is there any positive feedback for this?
Uh, this is the positive feedback reaction.
Okay.
And the three enzymes mainly are the regulatory enzymes, that is,
Mm-hmm.
I have mentioned over here.
But
That is
basically, whenever the sugar level goes up, at that time glycolysis starts, right? So, whenever there is excess sugar, it would be like negative thing.
okay. No, no, not an
Oh,
excess.
okay. All right. So there are three enzymes which I can see here: hexokinase, phosphofructokinase, and pyruvate kinase. Can you elaborate on them?
Yeah, hexokinase, since it is produced in liver,
Mm-hmm.
yeah, so it checks the level of glucose 6 phosphate,
Mm-hmm.
and in the excess condition, it inhibits the formation or production of glucose 6 phosphate.
Okay, since it is an
So,
irreversible reaction,
so but
that is
yeah,
the reason, right?
yeah,
Hmm. Okay.
and,
What about PFK?
it is again most regulatory enzyme, we can say.
Mm-hmm.
It
And PK?
regulates pyruvate kinase. Is it is a kinase, so the addition of phosphate groups takes
Okay.
place.
Okay, this is the
This is the
conventional thing which we see in our textbooks.
textbooks. Yeah, that is the whole glycolysis steps we can see over
Which
here.
we just discussed break by minor breakdowns, right?
Yes,
Here you can all view that how exactly glucose is getting converted into pyruvate. Two molecules of pyruvate is being formed, right, Miss Simran?
yes,
That two
Th-
molecules is because of this fourth step, maybe because two molecules are two, three carbon molecules are being formed, and that leads to formation of two molecules of pyruvate or something.
pyruvate.
Twice
Yeah,
it is happening in a cycle.
yes,
So it's done, I think now.
yes.
I said
So,
thank you.
no, since this was an overall gist of the glycolysis, I hope that students find it useful
Hmm.
for their preparation, and all the best wishes to them.
Yeah, it was really wonderful session, Miss Simran Motkekar. It was awesome. You joined us and shared your knowledge and you gave your time. To be very honest, time is very valuable, and people really think a lot to give their time. And I really thank you for joining us, imparting knowledge, making glycolysis simple, and what to say. I am really happy that we got time for today's session. And this is the segment under Rendezvous with Scientific Minds. So since it is something related to science, so what are your viewpoints on science as a whole?
Since a long time, I'm fascinated
Exactly.
actually, because science is something that we can see every day,
Hmm.
and we have to, if there is a thing, it's impossible that it's not proved in science. It's even the discoveries that are taking place and the research work that's ongoing, it's all about science. Means not exactly, science is a whole thing. There are
Hmm.
various fields that we have to explore, keep
To
exploring
put it very
and
simply, we cannot compartmentalize science
study
like
science.
maths,
Yeah,
like biology. Like even in biology, people bifurcate zoology, botany,
it's
biology.
important. Yeah.
That is so, like, we should interconnect things, don't you think?
Yes, it is a good idea.
Obviously, it should be because if we don't connect with things, then we won't get a logical reason out of anything, right?
Yes, yes.
And if it is not logical, and people won't be very much fascinated with science. Don't you
Science.
think?
Yes, yes, exactly.
So, how was
No,
your experience, Miss Simran? You can just share and we can.
yes, I would like to thank you for
Oh.
conducting such a, wonderful session for me, and I'm glad that I was a part of it. And I am thank you. I'm thanking you, and I'm also sorry if I missed out anything.
No, no,
And
I don't think so because we try to talk everything here and
yeah,
we try to make it simple as
simple
for
as
students.
yes, yes. Okay,
Okay,
so
Miss
that's
Simran,
all.
it was
Thank you.
lovely talking to you. This is your host, Miss Pillai from Rendezvous with Scientific Minds. Thank you so much, everyone. Take care. Signing off.